Exam tomorrow and erythropoiesis still feels confusing? Don't worry. In the next 5 minutes, we're going to revise the entire chapter using flowchart, diagrams, Viva points, and exam recall trick. No lengthy theory, no textbook paragraphs. Only the points that can help you score marks in MBBS physiology exam. So keep a notebook ready because I will show you the one-page exam recall sheet at the end. Before revision, remember the important thing. Examiners don't give marks only for knowledge, but they give marks for presentation. All examiners scan for these exact headings, so underline them. Starting with the definition. Keep it textbook accurate and concise. Introduction and classification. Show breadth of knowledge immediately. Main explanation. Use subheadings, no huge paragraphs. Flowchart and tables. Use contain complex pathways or comparisons here. Then diagram should be on the right side of the page in neat box with pencil labeled. Then clinical importance, the topper's secret weapon. And lastly the conclusion, one or two lines summarizing the impact. If you follow this structure consistently, your answer immediately look more organized and examiner friendly. Now let's revise the entire chapter under 5 minutes. Let's start with the most common mistake the students make. Mistake number 1. Writing the factors required for erythropoiesis randomly. Instead, remember them in categories like general factors, maturation factors. Mistake number 2. Writing the whole answer in a crowded fashion. No structure, no hierarchy, exhausting to read. You can fix them by keeping white space, bold subheadings, bullets, underlined keywords to get the maximum marks. In the next slide, I will show you the easiest flowchart to remember the entire maturation pathway. This single flowchart is enough to write the maturation process. So repeat after me. First, pluripotent stem cell, then colony forming unit GEMM, that is a myeloid series. Then come the burst forming unit erythroid. After that, colony forming unit erythroid, and then the maturation process, that is proerythroblast, then normoblast stages, which include 3 stages, that is early, intermediate, and late normoblast. Then comes the reticulocyte, and lastly the mature RBCs. This maturation process takes about 7 days. If you can reproduce this flowchart in exam, you have already secured a large chunk of marks. Now the favorite Viva question. Where does erythropoiesis occur throughout life? Remember this timeline. Mesoblastic stage yolk sac, hepatic stage liver and spleen, and myeloid stage bone marrow of exoskeleton and end of long bones. Simply remember yolk sac, liver and marrow. Embryo to adult in one line. This is one of the highest yield Viva question. Now let's quickly understand what happens during maturation. As you can see, these are the stages of erythropoiesis. Throughout the stages, cell size keeps on decreasing. Whereas in nucleus, initially it was present, but then eventually it also becomes smaller and then disappears. Whereas hemoglobin, it first appears in intermediate normoblast stage, and thereon it keeps on increasing. So these are the key changes that takes place in process of erythropoiesis. In short, RNA decreases, and finally the reticulocyte loses its residual RNA and becomes a mature RBC. In one single sentence, RBCs become smaller, more hemoglobin, no nucleus, and that becomes the mature RBC. Next comes the most important regulator of erythropoiesis. This flowchart is extremely important. Low oxygen here detected by kidneys, in turn which releases erythropoietin, which acts on the long bones and stimulate release of more RBCs. Remember, that's the entire story of erythropoietin. Hypoxia detected by kidney, which releases erythropoietin, which acts on bone marrow and produces more RBC. Draw this flowchart and examiner instantly know that you understand the concept very well. Students often forget this part of the answer, that is the factors required for erythropoiesis. Let's simplify. First one is the iron, which is required for hemoglobin synthesis. Next, vitamin B12, extrinsic factor for DNA maturation. Then comes the folic acid, active THA for DNA synthesis. And then the proteins for globin chains, and lastly the hormones like erythropoietin, androgens and thyroxine. You can remember these factors required for erythropoiesis by this mnemonic VIF, very important factors, that is V for vitamin B12, iron and folic acid. That's all you need for a 5-mark short note. Now regulation of erythropoiesis. This is simply a negative feedback loop. It starts with hypoxia, that is low tissue oxygen. It stimulates the kidney to release erythropoietin, which in turn stimulates the bone marrow and increases the RBC production, which improved the oxygen delivery, and thus the erythropoietin release is decreased. Now we can see the chronic hypoxia is the most common cause of polycythemia, and the cycle get completed. So whenever you see regulation question, draw this loop. Since you have stayed till the end of this video, I will give you 10 Viva questions. What is erythropoiesis? It is development and maturation of RBC. What is reticulocyte? These are the RBC precursor with RNA remnant. Where the erythropoietin is produced? It is produced in epithelial cells of the peritubular capillaries of the kidney. What is BFU-E? It is burst forming unit erythroid. What is CFU-E? It is colony forming unit erythroid. Normal retic count 0.5 to 2%. Total maturation time 7 days. 5 days to retic and 2 days to mature. Stain for reticulocytes. Brilliant crescent blue. When does the hemoglobin appear? Intermediate normoblast. When does the mitosis end? Intermediate normoblast. Now pause here and test yourself. Just before going for exam, remember this page. Sites, yolk sac, liver, bone marrow. Cells, stem cells, colony forming unit, blast, normoblast, reticulocytes and RBCs. Trigger, hypoxia, stimulate kidney to release erythropoietin which act on bone marrow and produces RBCs. And requirement for erythropoiesis, iron, vitamin B12 and folate for normal RBCs. Remember, intermediate normoblast is the stage where hemoglobin starts and mitosis stops. Reticulocytes contain RNA stained by the brilliant crescent blue, and kidney is the main source of erythropoietin. This one page can help you reproduce the entire answer. Now final topper steps. Always prefer flowchart over the text. Wherever possible, draw tables. And draw a neat and labeled diagram for erythropoiesis. And at the end of every physiological answer, you should write clinical correlation. And never forget to write all of these under a proper headings. These small presentation changes can significantly improve your score. If this revision helped you, watch the full 10 marks answer video on erythropoiesis next. And if you want to memorize the entire topic in just a few minutes, watch the memory trick version link on a screen. All the best for your exams.